All right. Good afternoon, everyone. Thank you for joining us today. My name is Alyssa Guido. I'm Program Director for the Pacific AIDS Education Training Center, Arizona, based at the University of Arizona College of Medicine, Tucson. Before we begin, I want to thank the telemedicine program for hosting our webinar today. Just a heads up, at the end of this webinar, you'll be directly taken to an evaluation following the webinar. That's for the telemedicine program, and you will also receive an email for an evaluation, another brief evaluation. That's for our AIDS Education and Training Center program. If you would be so kind to complete both evaluations, we'd really appreciate it. Those are required evaluations for our funding. Just to let you know, please enter your questions into the chat throughout the presentation today. We'll be monitoring the chat and we'll let Dr. Branson know when questions come up. So with that, I'll introduce our honored guest today. Dr. Bernie Branson has done HIV testing for over 35 years. He wrote the CDC 2006 HIV testing recommendations and then the 2014 HIV testing algorithm. He served as the CDC Associate Director for Laboratory Diagnostics for 11 years prior to his retirement in 2014, and currently provides expert consultation and training on HIV and COVID-19 diagnostics. And with that, I'll turn it over to you, Dr. Branson.
Great, thank you, Alyssa. It is nice to be here with you today. I'm sorry that I can't be there in person to speak in Tucson. It's always was a favorite thing of mine to do, maybe now post-COVID. So what I'm going to talk about today is mostly COVID-19 testing and somewhat about at least the new developments in HIV testing. To start off with, this is the disclaimer from HRSA that says they are not responsible or have not endorsed anything that I have to say. And this is supported by the Pacific AIDS Education and Training Centers, and that is funded by HRSA. I'll also, in my presentation, refer to some individual tests by brand names. That's for the purposes of endorsement, for identification. And for clarity, I don't mean to endorse any specific tests. Our objectives today for this session are to understand the fundamentals of laboratory tests for SARS-CoV-2, appreciate the role for antigen, PCR, and serology tests in identifying exposure to SARS-CoV-2, review new developments in HIV tests and the utility for a new diagnostic algorithm, and finally describe how similar parameters affect the accuracy of tests for SARS-CoV-2 and HIV. And you notice in this objective slide, I talked about SARS-CoV-2 a lot, and I may slip and go back to talking about COVID-19, but in general, SARS-CoV-2 is in fact the name of the virus, and COVID-19 is the name of the disease that's caused by the coronavirus that was identified in 2019. So that's the sort of difference between those two. We have a couple of poll questions to ask you today, and the first one is this: which COVID-19 tests have received FDA approval? PCR tests, antigen tests, antibody tests, home tests, all of the above, or none of the above? So I want to see pretty much how much folks already know about this topic. If you'd make a selection and complete the poll, and I think that will soon bring up the score. Guess I don't have to you do something about that, right? About ending the poll. So the winner among the current audience is all of the above, and then some smattering of people thinking antibody tests, antigen tests, and PCR tests. About 24% of people got the right answer, which is none of the above. In FDA regulation of devices, just to give you a quick primer on this, there's a license, first of all, for biologics. Tests that are used for blood screening are biologics, and so HIV tests that are used for blood screening and for screening blood supply, like the ELISAs and the are licensed. Tests that are for high-risk devices classified by the FDA as such require approval or get approval, and they have to submit a pre-market approval application. And this again is HIV antigen, antibody, and RNA tests. The HIV antigen antibody test, in fact, is not licensed because it's not used for blood screening. Most diagnostic tests that laboratories use all the time go through what's called clearance, not approval process. and they get put in a 510K application, which is much easier, shorter, and less expensive. And finally, what tests have now is emergency use authorization, and that's not approval. I want to talk a little bit about that. There's a few more regulations that apply to tests. One of them is CLIA, the Clinical Laboratory Improvement Amendments. Part of CMS, the Center for Medicare and Medicaid Services, classifies devices and certifies labs to perform tests based on how complex they are. And there's 3 categories: high complexity, moderate complexity, and waived. And home tests are automatically waived tests. Then there's another category called laboratory developed tests, for which at the current time, these LDPs, FDA requirements, have been suspended. And they are for use only by the laboratory that developed the assay itself. It can either be a novel assay, like the new ones that came out for SARS-CoV-2, or it can be an assay for which other assays already exist, for example, vitamin D. And the difference in those categorizations, FDA approval versus cleared versus emergency use authorization, give you different degrees of confidence in the results of tests. So that here, for example, we're looking at these categories of tests. And so the Aptima HIV RNA test and the Bio-Rad antigen antibody EIA test, when you look at the number of specimens that were used in order to get approval, over 1,000 positives, over 3,000 negatives, and they have very high sensitivity, very high specificity with very narrow confidence intervals. The confidence interval is an indication of basically if you reran the same experiment 100 times, 95% of the time the result would come out between 99.6 and 100 in this case. The FDA cleared tests, like the, for example, the influenza test, the cobas flu A and the flu B, there are RT-PCRs, which we'll talk more about. But as you see, to get clearance, they only have to test 175 positives, 42 in case of influenza B, and about 1,000 negatives. And consequently, the sensitivity and specificity of those tests are by their nature lower. And because of these smaller numbers, the confidence interval is much wider. So the sensitivity could really be as low as 95%. And even though the point estimate here is 95.2%, it might really only be about 84% sensitive. Now, flu is a little bit different because flu changes a little bit every year, and so the flu tests have to be repeated or rejiggered every year to detect the antibody that's out there. Now, if you're looking for the rapid test, the Binax Now is a flu antibody test and also a flu B test. These are both antigen tests for flu. And again, you see the number of positive specimens were 69 and 50. The number of negative specimens are 200, around less than 300 in their cases. And again, they have relatively low sensitivity and specificity. These are the, rapid antigen point of care tests. And now we get down to the emergency use authorization test here. And what you see is from the Abbott SARS-CoV-2. This is the Abbott PCR, one of the earlier ones that come out. They again tested 84 positive specimens, 120 negative specimens, and it showed good sensitivity and specificity, but the confidence interval, the range of what that might be if you tested it on more people, is really pretty wide. And similarly now, the rapid antigen test, this is the Binax Now, was the one that was being used to screen a lot of the visitors to the White House before the super spreader event. And you see that it basically, they got their authorization based on testing 35 positive specimens and 67 negative specimens. And their sensitivity was technically 97%, but the range for the confidence interval was quite wide, down to as low as 85% for sensitivity. So how confident you are in any one of these results really depends on what the number of specimens were required for the FDA to look at it in order to get it out onto the market. So all the SARS-CoV-2 tests come under what's called Section 336. I'm sorry, they have 336 so far emergency use authorizations. That's the number of tests that have FDA's okay. At the current time, there are over 200 RT-PCRs, 18 for saliva specimens, 19 for home swab collection systems, 15 for the COVID-19 influenza A and B multiplex test. So first, you take a respiratory symptom and you run it into this test, and it's either influenza or COVID-19. Sorry for the typo. That test will turn positive. I can't be sure because these numbers keep changing, but there were 16 emergency use authorizations for antigen tests. 8 were for high or moderate complexity lab assays. 7 of them were for waived rapid tests that are waived complexity. And one, in fact, is a home rapid test that is read by an instrument. And then there are about 65 antibody tests with emergency use authorizations. By comparison, there are, I think, 6 total HIV antigen antibody assays that have received FDA approval, and 1 differentiation assay and 2 of the RNA assays for diagnostic uses. So to talk about what the tests are, first of all, I'm going to talk about what the RT-PCR is. It's the one for SARS-CoV-2 RNA. This is an RNA virus. And there are a series of steps that go into doing the test. First one is tissue sample. And the sampling method depends on what it is. Looking at this, they did look at bronchial lavage, nasopharyngeal swabs, throat swabs, sputum tests, saliva tests, and the bronchoalveolar lavage, looking at 1,000 specimens from 200 patients, picked up the virus in 93% of them, that nasopharyngeal swab, 63% of them, the throat swab, 126, or only 32% by doing a throat swab. Sputum specimen was actually pretty good, 72% in this. And in fact, people also excrete the virus in their feces, and we're able to test for that and pick it up in about 29% of the specimens that were tested. So after the sampling stuff, you do nucleic acid isolation. And what that means is that you want to run this test only on RNA, the viral RNA. And so you want to get rid of the cellular debris. You want to get rid of any DNA that's there, that's part of the host, that's part of the reaction. And then you use the enzyme reverse transcriptase to form what is called complementary DNA. RNA, as you know, is a single strand. DNA is a double strand that's hooked together. And so the reverse transcriptase takes RNA and turns it into a DNA, which allows this whole PCR thing to happen, at which time you do the real-time DNA PCR by using the complementary DNA made from that piece of RNA. And what happens basically is that usually requires RNA extraction. And so the extraction requires a swab, and it requires the preservative in it that is good for DNA and RNA. And then you have to collect the sample, and you go through a bunch of steps, adding alcohol, transferring it to a column, and centrifuging it down, and doing a bunch of other things with centrifuges and a wash buffer and a new tube and back and forth. And this extraction kit thing was one of the shortages that really beset the early HIV testing kind of thing. And then there were shortages of swabs and preservatives. And so the things that led to the problems with testing were because it's a complicated procedure. The procedure itself for RT-PCR starts out with this double strand, like I mentioned, the complementary DNA strand up there, and the reverse transcriptase makes that from the virus RNA. Then you use heat, and it denatures those 2 strands of DNA into 2 single strands, and you then use these primer things which match those single strands and treat that with an enzyme. And as soon as you lower the temperature again, you see this one now went down from 98 degrees down to 48 to 72 degrees. The reverse transcriptase works along those strands, and they keep adding to the chain, making 2 new complementary DNA molecules. And then you start from scratch, take those, separate each of those 2 molecules again, raise the temperature to denature them, lower the temperature to create new ones in that regard, add the DNA polymerase, and repeat it. Each one of these things, which causes for heat and cool and heat, is called the thermocycle. And so these PCRs depend, well, most of them, on thermocycling. And what that creates is a chain reaction. So in the first cycle, as you see, you took your original 2 copies and you made 4. And then in the second cycle, those 4 copies make 8. Third cycle, the copies make 16. And by the 30th cycle, you started with this, 2 copies, and you're at 2 billion copies by running 38 thermocycles through the PCR. And so that's how it amplifies a very small amount of specimen into something that then becomes large enough to be detected on an instrument. And 2 to the 21 is 1 million copies, 2 to the 31 is 2 billion copies. The deal is the number in the copies in the first cycle is determined by the number of copies in the specimen. So if you start with a bunch of copies in the first one, it really adds up very quickly. And so you get what's called a cycle threshold. So something that is high positive, something that has a lot of viral RNA in it, will multiply very quickly, and then only a couple of cycles, between 3 and 8 cycles, you'll get a positive test result. But because if it's only, let's say, one RNA strand in it, it may have to go on for a long time. There is a cycle threshold. So you have to keep cycling it, and that's usually around 30 cycles in order to be certain that it's negative. And you may have heard in the past that with many of these tests, they take longer when they're negative than when they're positive. And that's because of the number of cycle thresholds that you have to run through before you can say that it's negative. So looking at the comparative sensitivity of these tests, the FDA put out something that was called a performance panel and asked all the manufacturers who had gotten an emergency use authorization to run this performance panel and to compare it sort of head to head with the other tests, excuse me. As you can see, these are the data from the manufacturer, the limit of detection. Now, the FDA used something called NAT detectable units per milliliter, which is not exactly clear what that's supposed to be because it might not be all viral copies. But as you can see, they range from as low as 180 or 600 for the Perkin-Elman tests and for the hologic test to about 5,400 for the original. I'm sorry, this is a new CDC test, the influenza and SARS-CoV-2 multiplex assay. The first CDC test had a limit of 18,000. The Abbott Diagnostics, this is their COVID 13-minute PCR test, has a limit of detection of 300,000 of those copies. And then another rapid test that's been approved, the Live Direct, which takes only about an hour, has a limited detection of 500 to 40,000 copies. But you see that the longer the test takes, the better is its sensitivity. The quicker the test is, the less accurate the test is, or at least the lower is its limit of detection. So those there basically are looking at about 2.5 hours for just the laboratory processing, not for the extraction steps and other kinds of things. And then the rapid tests, as you see down here, are shorter. The CEPI is a SARS-CoV-2 rapid test that runs a little machine. And then these are the point of care tests, which are pretty quick. And what I want to do now is ask our second poll question, which is to compare saliva specimens with the nasal swabs and tell me which statement is incorrect. Saliva is less accurate than nasal swabs. Saliva is easier to collect than nasal swabs. What is detected, a virus, I'm sorry, is detected longer in saliva than it is in nasal swabs. Saliva specimens require special preservatives for RT-PCR, and RT-PCR is more expensive with saliva specimens because of special handling and extraction procedures. So tell me which one of those things you think is correct. I'm sorry, which one is incorrect? Don't vote for the correct answer, pick the incorrect answer. Wish we could see the voting as it was happening, but so we're probably ready to move on, I think. So the people who basically were all over the map with this one, and for the one that is incorrect is the last one, that RT-PCR is more expensive with saliva specimens because of special handling and extraction procedures. Because in fact, what it is basically, there's a saliva test that has an emergency use authorization. This was developed by the Yale School of Public Health, and the EUA was issued back in August for them. It uses unprocessed saliva that you just spit into a tube. It can be pretty much any kind of test tube, laboratory tube, blood tube. You add then something to it called protein ASK, and you mix it on a little mixer, and then you heat and activate it and subject it to PCR. And so it uses unprocessed saliva. It's collected into a sterile urine cup or test tube. No special equipment, no extraction step is required. And in fact, this was the one that was tested and proven to be beneficial during the whole NBA bubble thing last year. They were getting this live direct test, I guess, every day or maybe every 2 days. And what it basically does is it eliminates the need to collect a nasopharyngeal swab. So there's no swab, no sticking something up your nose, no transport medium. It eliminates the need for an RNA extraction kit, which, as I mentioned, for a while were the basis of supply issues. And Yale has validated it for use with reagents, primers, and instruments from multiple different diagnostics manufacturers. So the procedure is just their procedure that you have to follow. You don't have to use a special instrument or special reagents for it. It is distributed free to individual laboratories. Commercial laboratories have to obtain a license to use it. It's free, that license, but in the license, they must negotiate the retail price that they can charge for the test. Yale, puts the stipulation in place that if you're going to use their protocol, it seems to make sense to do so. They don't want them to overcharge for it. So I think it's a pretty decent test. And this is some of the early data that was published on it, looking at paired specimens from hospitalized patients who had NP swabs that were positive for SARS on their hospital admission. And what you see here is matching the nasopharyngeal swabs and the saliva swab samples. You basically get pretty much better detection with saliva samples than you do with the nasopharyngeal samples. And this is looking at the people testing positive nasopharyngeal samples and saliva samples over time from the time that they had come into the hospital. And you can see that the saliva actually tests positive longer in these cases than it does in the nasopharyngeal swabs. And in addition, looking at its stability without preservatives, it is stable, as you can see here, at basically 30 degrees centigrade or above for at least 7 days without any special handling. And this is comparison of doing an extraction step here with not doing the extraction step here. And you can see it did have a couple of them that fell out without the extraction step. But it was very accurate, easy to collect, easy to handle and transport. And the total minimum reagent cost per sample for the reagents alone, depending on whose you buy, ranges from $1.29 to $4.37. So I think I'd like to see a lot more saliva testing going on because it's easier to do with less risk for healthcare workers. And because there's no extraction step, it's also faster. Just to show you what it looks like, these are from the first US case of COVID, the CDC's electron micrograph, showing the blue things here, which are the virus. And you can see its viral genome here around the outside. So it's a pretty cool micrograph. But looking at the viral antigens and the RNA itself, as you can see, this is a sort of schematic of the virus. Sorry, I got muted. It's got a spike glycoprotein on the outside, and then a membrane protein, and then a nucleoprotein, which is its RNA inside the genomic RNA, and then another envelope small membrane protein, which are all characteristic for the SARS virus. The SARS-CoV-2 multiplex test from CDC detects the N gene, the nucleoprotein gene from SARS, the matrix gene for A, the NS2 gene for influenza B, and then there's an internal control and a positive control. And one of the reasons this is important is that a lot of the tests, or at least several of them that test for this, including the PCRs on here, test for the spike glycoprotein and not the nucleoprotein, the N nucleoprotein. And the variants have shown some of the PCRs for the spike to be negative in people who have positive. And in fact, the original ones that were coming out of Great Britain with the British variant that showed some tests didn't pick it up were called, they're classified as S negative PCRs because the PCR was positive, but when you do a multiplex, you can do it for more than one gene, and they were finding ones that did not have the S gene present in it. So in terms of testing strategies, that was sort of the first part we wanted to talk about, which is the laboratory basics of it. And maybe we'll stop for a second and find out. Alyssa, any questions so far?
We do have a question. I don't know if you can see it if you click on the chat button, but I'll go ahead and read it. Please repeat why you'd like more saliva testing done. Safer and cheaper aside, is it as sensitive / specific as NP, I guess that's nasal pharyngeal swabbing? Thank you.
They have a couple of slides on that. I'm going to wait to answer the question. But the answer, to be honest with you, is yes. One of the tests actually has been developed and used by a guy down at the University of Arizona. They've been very progressive in the stuff they've been doing, but His basically he has a little rinse that you have to rinse your mouth out because he says it's so dry in Arizona, people can't make enough saliva. So now we'll talk about testing strategies, which gets, I think, a bit to this point about sensitivity and specificity because it makes a difference. Testing, you can do the old CDC guidelines, and the revised CDC guidelines were for testing of symptomatic people only. You can test asymptomatic persons and known contacts, and especially you might want to do that in places with conjugate living, like dormitories and nursing homes and places like that. The big movement that has gone on, of course, is to test anyone on demand. And there were huge lines at testing places until the vaccine came out, and now there's huge lines at vaccine places. You can test everybody twice a week, like University of Illinois did for saliva. You can do focused testing after sentinel testing. At first, University of Arizona tested the sewage from specific dormitories there, and they were actually able to detect an outbreak of 4 asymptomatic cases by testing the sewage from an entire dormitory. So it was a pretty interesting approach to trying to find it. And that takes us to the screening strategies for COVID. And interestingly enough, they showed the test sensitivity is secondary to the frequency that you get tested and the turnaround time for the COVID results. And I'm going to go through that a little bit because we used to always think we want the most sensitive test possible. So looking at a conjugate setting, this is the thing that was modeled out in that paper that I just showed you the title of. You see here that here's a limited detection of 10 to the third, which is 1,000 copies. Here's a limited detection of 10 to the fifth, which is 10,000 copies. And you look at basically the difference in how long it took the test to pick it up. And this picked it up at about 28 hours, and this picked it up at about 32 hours after the time of exposure. This is viral load that you're looking at here on this graph. And this is when the positive tests and the diagnosis of COVID were returned. And that's because of the delays with PCR and stuff. You could pick it up very early, but you don't get the result back for some period of time. So the number of infections that you identify in quarantine depends more on the frequency that you're testing than on the test sensitivity. Because if you're not testing very often, you're going to miss people when they first become positive. And so this was sort of the modeling of basically how many people would end up being identified and quarantined as soon as possible based on either symptoms or on periodic testing. And the pink is the pink is the more sensitive test, and the brown color here is the less sensitive test. So about 25% of people who get symptoms would end up identifying and quarantining, and so no tests, they would still be found. If you test everybody every day, you pretty much find every infection. And if you test everybody every 3 days, you basically pretty much find over 80% of the infections. And there's not much difference between the less sensitive and the more sensitive tests. Now, when you test people every week in that kind of situation, you only pick up around half of the folks and quarantine them in time. And testing every 2 weeks is about the same as not testing them at all. Well, they're still self-isolating here, so they still get symptomatic. But again, the total symptomatic people plus tested people here is only coming up to about 45% when you're testing it for 14 days. So obviously, it seems that if you want to sort of balance off turnaround time and costs and things like that, this looks like twice a week or every 3 days probably is going to be your best bet. Now, there's some other stuff that's going on. The RT-PCR is exquisitely sensitive for the presence of viral material. And most tests report their sensitivity compared with the RT-PCR. It's sort of considered the gold standard. The RT-PCR, however, also detects viral transcripts that are fragments that cells produce in excess compared to the number of actually infectious virions. And looking at a review now, a meta-analysis, infectious virus is very rarely present at RT-PCR values of less than 10 to the 6th copies per milliliter. That's 100,000 copies per milliliter. If it's less than that, infectious virus is usually not there. It's usually positive from viral fragments. And those of you who are familiar with doing testing after chlamydia treatment are familiar with the fact that the PCR, the genetic probe, stays positive for long after the infection itself is gone, not because there are infectious agents still there, but because of the fragments that have been produced. Now, antigen tests versus RT-PCR tests. The antigen tests are visually read antigen-antibody interactions. They're not all visually read. As I mentioned, one of them has a little reader app that it uses. Antigen sensitivity is not really sensitivity, because what they're comparing it with is the RT-PCR. And so what you get is positive percent agreement with the RT-PCR, which is not necessarily the same as sensitivity. Because when they looked at paired swabs from college students, there was an overall prevalence of 5.2% in this group. 80% of the people who had symptoms had a positive PCR test. 41% of the people, and most of the people were asymptomatic, had a positive PCR antigen test when the PCR test was positive. So you see that in the symptomatic people up here, probably had higher viral levels, the antigen performance was really good. Among the asymptomatic people here who may have had lower levels of virus, the antigen performance was a lot lousier. Now, there's also a whole study done by the UK in Oxford where they looked at the sensitivity. This is of a rapid test, rapid antigen test, and the test compared to PCR was 79% when performed by lab professionals, 73% when performed by trained healthcare workers, and 57% when performed by self-trained public using a protocol. So these are all the same tests, but the sensitivity vary depending on who's doing it, whether people had symptoms or not. There's a bunch of things that contribute to what the value of the test is. Now, no study has identified infectious virus in people more than 9 days after their onset of symptoms, despite weeks of persistently positive PCRs. RT-PCRs have limits of detection of between 10 to the third and 10 to the fourth copies per milliliter and lower, but the infectious virus is very rare at RNA values less than 10 to the sixth copies, a level that antigen tests can achieve. So genetic variants might lead to false negative RT-PCRs, excuse me, if the mutation involves part of the genome that is detected by the test. So the role for antigen tests is they take 15 to 20 minutes. RT-PCR takes about 4 hours. Antigen tests are less expensive and suitable for point of care. Antigens likely detect persons who are infectious, but they might be false negative in some cases. And the RT-PCR remains positive long after infectious virus is gone and the antigen turns negative. The role for antigen tests makes repeated serial testing positive often enough because it delivers results fast enough at costs that are low enough to make surveillance testing feasible in dormitories, schools, nursing homes, job sites, that kind of thing, where you can test people periodically in order to identify those who are infectious as soon as possible. This was a huge area of debate recently until we did come out with the vaccine because, for example, in nursing homes and other places, you wanted to isolate, get people done as soon as possible. And so this was, that paper again, the COVID-19 screening strategies, looking at frequency and turnaround time. And I want to mention the home test. This is a LU home test for SARS-CoV-2 antigen that was developed in Australia. It's got an emergency use authorization in the United States. It's got, the way it works, it's got a flip top lid with a swab tip and a little child adapter so you don't push it too far up into the kid. The processing fluid, and you put the specimen into the processing fluid with the dropper on top, and then the test has got a sample port and it's got a button in order to analyze it. This again uses nasal swabs. And it's a 15-minute fluorescent antigen test. It has an app for iOS and Android. And when you sign into the app, you have video and step-by-step instructions on how to do it. And then the analyzer is battery-operated fluorescent analyzer, which connects with your phone, and the results show up on your phone after 15 minutes. And it also automatically the report to the health department, reports the results to the health department, and it'll email you a result but that's optional. So this is the home test of nasopharyngeal swabs, and I think that there are several promising ones as well, looking at potentially the use of saliva for that. The Department of Defense actually awarded this company $238 million to increase their domestic production capacity and deliver COVID home tests for people. This is done back in February when the Biden administration made the announcement of the big purchase. So now my next question is, and sorry, I did not, well, I'll get to the question first as you put the poll up. The third poll question is, what statement is true about antibody tests? Positive antibody test means the person has COVID-19. A positive antibody test means the person is immune. Does a positive antibody test provide insight into the prevalence of COVID-19 in the population, or do they help identify new mutant viral variants? I want to make a selection from that group. And again, we're looking for which statement is true. So let's have a look at the poll results. So this, everybody's right on target. It provides information on the prevalence. It does not necessarily mean that a person is immune. You have to have neutralizing antibodies in order to be immune. And as we have now seen with some of the new variants with the different spike protein on top, people can get reinfected by a variant that differs from the one that induced their antibodies in the first place. So now PCR versus antibody tests. PCR tests detect the presence of viral material, but they can't determine whether a person has been infected previously and recovered. And sometimes, as I mentioned, they continue to detect virus after the infection has resolved. Serology tests determine whether a person has been previously exposed to a pathogen. They provide insight into the prevalence of the disease in the population by identifying people who have specific antibodies. So I didn't answer the earlier question, which I want to get back to about the saliva versus nasopharyngeal swabs. There have now been several studies that show as a specimen type, saliva is more sensitive than the nasopharyngeal swabs. So even there are several saliva tests that aren't the Yale method. They do extraction steps on them with their PCR, but the saliva looks like it's actually a more desirable specimen, and I'm not quite sure why people haven't moved in the direction of using saliva a good bit more, because it's easier, faster, and sure better than sticking something up your nose. So I now want to talk about a little FDA regulation stuff again, looking at lessons from HIV testing. With HIV, the antibody tests came first, and they were intended for screening the blood supply at the time. So the test received a high-risk designation by the FDA, which required PMA, pre-market approval. And the part of FDA that handles this is the Center for Biologics, who usually handles vaccine approvals and blood screening test approvals, which things are being used in making recommendations for blood donors. CDRH, the Center for Devices and Radiologic Health, is the part of FDA that regulates and evaluates almost all other tests, except for high risk tests designated like that. In HIV, the first quantitative HIV-1 viral load monitoring test wasn't approved until 1996, about 11 years after the first antibody test. And the first HIV-1 RNA diagnostic test, the other quantitative tests are only approved for monitoring. The first one approved for diagnosis was in 2006, which was the Aptin test. Just as an aside, the FDA, both CBER and CDRH, proposed down classifying HIV and HCV tests to class 2, which would make them comparable to most other tests. It was recommended by an advisory committee in 2018, and they had public comment period in the spring of 2020, and so it's only 3 years later now, and we're still waiting. FDA required COVID-19 laboratory diagnostic tests to obtain emergency use authorizations at first. Then they granted blanket emergency use authorization, first to the LDT PCR tests. It first granted a waiver from EUAs for antibody tests, and also okayed CLIA waiver on antibody tests. So they did not have to come in for EUA. But they did end up issuing EUAs for several antibody tests and rescinded them later and started requiring EUAs for the others when they found out that they weren't very accurate. So that's a whole lot different than HIV. FDA has so far rescinded 254 EUAs for COVID-19 tests. So when you get your COVID test, you never know quite what you might be getting. There are different types of SARS-CoV-2 antibody tests, lateral flow assays, ELISA tests, neutralizing antibody assays, so that the lateral flow assays are usually antigen tests, usually a rapid point of care tests for antibody. The ELISA tests are like other ELISAs, chemiluminescent, electrochemiluminescent. They have the potential for lots of testing, high throughput. And then the neutralizing antibody assays are serial solutions of serum added to viral cultures to look for viral inhibitors. So this neutralizing antibody assay is the one that looks basically for immunity that is present in people. And because you have to add them to viral cultures in biosafety labs, they are not very widely available. So looking at antibody in people with COVID-19, the antibody is dynamic. It changes in the IgG levels. This is based on observations from 285 patients. 100% of them had tested positive for antibody within 19 days after their symptom onset. So that's within 3 weeks. The seroconversion for IgG and IgM occurred simultaneously or immediately after each other, unlike in HIV where IgM precedes IgG appearance by 5 or 6 days. Positive HIV antibody tests, of course, indicate that you have active HIV infection. Everybody who's antibody positive has HIV. Positive COVID-19 antibody tests, first of all, a single test has a low predictive value in most populations. It is indicative of past exposure, not necessarily active infection. It's not known whether antibody tests indicate immune protection, and if they confer immunity, it's not certain how long antibodies will persist. There's still a bunch of unanswered questions about antibody tests and immunity. Now we get to the bugaboo that everybody really dislikes, and I can't seem to get myself unmuted.
We can hear you.
Sorry, I'm confused by the little icons up there. So for interpreting test results, sensitivity and specificity, we talked about some. And sensitivity technically is the probability that the test is positive if the patient is positive. Specificity, the probability the test is negative if the patient is negative. Now, the predictive value is what we really are after, which is the probability the patient is positive if the test is positive, or that the patient is negative if the test is negative. For COVID tests, of course, you're really talking about positive percent agreement, which is probability that one test is positive if the other test is positive. In most cases, antigen test if the PCR test, or obviously one PCR test compared to another PCR test. And similarly, it's not quite specificity, but negative percent agreement, which is probability that the test is negative, the second test is negative if the first test is negative. So what we really want to get to is whether the patient is positive or not, and that's a lot tougher. So looking at the SARS-CoV-2 antibody test, this is 108 blood donor plasma specimens collected before July 2018, so there was no SARS-CoV-2. So everybody should be negative here, right? So as you can see, the mean specificity, or negative percent agreement, but the specificity here, because it's always PCR, was about 94%. And so you see that depending on which test it is, anywhere from 0 to 17 of the tests were positive in people before COVID-HIV-2 was invented. SARS-CoV-2. So now, my fourth poll question, the one everyone always hates, what happens if results of tests when the prevalence of disease is low? Is the number of false positive tests higher? Is there a higher likelihood that a positive result is a false positive? Is the chance of false positive results being reduced by low prevalence, or is basically nothing? False positive results are unaffected by prevalence. So I submitted my answer. We'll be ready for a couple of minutes to post it. Okay, Kristen, we're sort of ready. So the higher likelihood that a positive result is false positive. This is a very, very smart group for people to know that distinction. We'll go through the example quickly anyway. We're going to test 10,000 people, and we're going to use a specificity of 94.2%. That was the mean, as you saw, of those PCR tests from that study. So 58 out of 100, I'm sorry, 58 out of 1,000 people will be false positive. That's what this specificity means. So that, 4.2 is 5.8% of them are going to be false positive. So looking at a place that has high SARS-CoV-2 prevalence, like Arizona was when they were in the throes of the epidemic, let's say 10.5% among people tested. If we tested 10,000 people, then with 10.5% prevalence, we'd expect 1,050 people to be true positive. And because of this specificity, we would expect 580 people to be false positive at that prevalence. And so the positive predictive value there is 1,050 out of those 1,630 total positives, or 65%, which is pretty decent. I mean, at really high prevalence like that, you can sort of afford a test that doesn't have the greatest specificity in the world. So now, basically, we've gone through that experiment and showed pretty much well how it worked. Now we're going to do it in a place where the SARS-CoV-2 positivity is only 1.3%. Like it was in a lot of New York cities when they started to open some of the schools. In that case, if we test 10,000 people, there will be 130 true positives. And because we're using the same test, they're going to be 580 false positives, the same as before. So that in this situation, the positive predictive value is only 18%. Only 130 out of those 700 tests are positive. And so as you can see here, number one, the number of false positives hasn't changed. It's the same in both situations with the difference in prevalence. When the prevalence goes down, however, the number of true positives goes down. And so the likelihood that a positive result is false is higher when the prevalence goes down. The difference, between that positivity and the other positivity is substantial there, bringing the number of true positives down. If we now use a test that's got better specificity, there would only be 15 per 1,000 that were false positives. Then, testing that same low prevalence group, we see there's 130 true positives and only 150 false positives. So it's better, at least it's only about 50-50 as opposed to 18%, like we saw before with the lower specificity tests. That's why people try to shoot for tests with high specificity. The point, though, here is that a lot of these positive tests, are, depending on the prevalence situation, likely to be false positive. And there are not many tests that get up to the super high 98% specificity. So to talk about HIV for a minute, because this is part of the AIDS Education and Training Center network. This is HIV virus, and we're all familiar with the markers that GP120 and GP41 are outside of the membrane. P24 is the antigen on the inside. The molecule of RNA is what the virus is actually made of. And then there's a gene called the POL gene, which codes for protease and reverse transcriptase and polymerase and integrase, all the things we have our drugs to work against. So this is the current algorithm, diagnostic algorithm. Let's start with an antigen antibody screening assay. And then you go down to an HIV-1, HIV-2 antibody differentiation assay. And if that is indeterminate or negative, excuse me, you'd want to do HIV-1 NAT. Now, until now, the only test that was approved for diagnosis for use in that algorithm was a qualitative assay. It means it gave you just a positive or a negative. In November of this year, the Aptima HIV-1 quantitative diagnostic assay received an approval for both diagnosis and for monitoring of viral loads. So if you would take this algorithm here, you could use that NAT to resolve negative or indeterminate results. The Aptima dual claim, as I said, is for diagnosis on serum or plasma. You can use either specimen for diagnosis. For monitoring, it can only be done on plasma. The plasma accurate is the only one that is comparable to the result that was done on plasma samples for all the clinical trials and circumstances like that. In reality, the serum value is within 1 log, within 1,000 copies of what the plasma value is. Usually it's lower because some of the virus gets caught up in the clot when you the blood clots with serum. This is actually looking at that sort of comparison of specimens. This was done from the Florida Public Health Laboratory when they were comparing their qualitative specimen on plasma with the qualitative on serum. The qualitative on serum and the specimens. Basically, what you see here is that the qualitative test on plasma was non-reactive in 3 cases, and on serum, it was non-reactive in 4 cases, and all of them were at really, really low levels of virus, less than 1.47 log, which is less than 150. Well, actually, that's 32 copies. And then here, when we're up to 300 copies already, basically everything is reactive on serum or plasma, so that you can get copy numbers. The system produces them on either specimen type, but if you're using it for monitoring and for periodic follow-up, you have to use plasma specimens. Sorry, this isn't there twice. Well, so now, though, I'm proposing that for what makes sense at the current time for the new algorithm is that we do the antigen antibody assay, and then if it's positive, we go directly to a viral load. We don't need to go to a differentiation assay anymore, because if the viral load is detectable, you treat somebody. And these days, of course, you want to try to get treatments at the same day if you can. And it doesn't make sense to wait for another antibody test result, because with the specificity of these antigen antibody immunoassays, 99.6% of the people who are positive on the first test are going to have detectable RNA and need treatment. If it is undetectable in those initial specimens, then you can go on to do the antibody differentiation immunoassay. And it'll tell you whether it's HIV-1 or HIV-2 based on the antibody test. In general, this would pick up the vanishingly rare cases of HIV-2, and it would also pick up the people who end up getting tested while they're already on antiviral therapy and they have undetectable virus because they're virally suppressed. And so those people will test negative on the viral load test. It has a very low limit of detection, 60 copies. And they would still be, in most cases, antibody positive and HIV infected. And then if they were negative or indeterminate on this second one, on this assay, differentiation assay, especially if they're indeterminate, you then would probably go on and do either second different antigen antibody assay or an additional NAT. But basically, most people, really 99.6%, would be taken care of by doing this viral load as the second step. And it would basically cut out one whole test, which is the extra antibody assay that we're doing on everybody right now. And it would give you immediately clinically useful information, knowing what their viral load is. To be honest, the precision of the viral load is less important at the time of initial diagnosis, because there is a threshold for one antiviral drug. If the viral load is greater than 100,000, you're not supposed to use it. And there's a second one you're not supposed to use if the viral load is greater than 500,000. And this test will tell you that range on serum or on plasma. That's the quantitative viral load assay. And I'm hoping that will really foster the availability of point-of-care nucleic acid tests. If you're the second test in the, nobody wants to be the third test in the algorithm, right? Because everybody gets the first test, everybody who's positive gets the second test, and only the occasional problematic specimen or the acute infection kind of thing goes on to get the third, the NAT test. And by that time, you've introduced a substantial delay before the result comes back, because so many of those qualitative nucleic acid tests are done. So few of them are done, rather, the qualitative tests. They're not available in a whole lot of laboratories, and the company has also issued the end-of-life announcement for the qualitative nucleic acid test. So it's going to be off the market in a year, and the available supplies that are shipped during the next year will expire a year after that. So we'll only be here for 2 more years, and then we'll be going to the quantitative test. In this one, which is a gene expert, and some of you, if you have any hospital background, you may be familiar with, many hospitals use this to test for MRSA and to test for resistance for tuberculosis. There are a number of PCR tests that run on this platform. It can be scaled up to multiple little things like this, or used one at a time. Point of care, it takes 1 milliliter of plasma, results in 90 minutes. Limited detection is 32 copies. It's been available in Europe since 2014, so we're a little slow here in this country. So in summary, the regulation of COVID-19 tests is substantially different than that for HIV tests, and it shows. RNA and viral load, I think, are going to play an increasingly important role in HIV diagnosis in the future. And if RNA tests get cheap enough, we could probably use it as the first test instead of the antigen antibody test. Most COVID-19 diagnoses depend on PCR testing of nasopharyngeal swabs, but saliva specimens offer an attractive and potentially more sensitive alternative. Antibody testing is going to help identify people who have already been infected And serial antigen testing, especially if it can be point of care or optimally home testing, might really play a useful role in limiting COVID-19 transmission. So that's bringing us close to the end of our time. And I did want to mention one other development. This is Ohio State's COVID-19 breathalyzer test that's still waiting FDA approval. There was, I should say authorization. It was submitted in January 25th of this year for authorization. I was showing this as we went through a little practice session because Alyssa had to go through my slides ahead of time. She thought it was, she said, okay, what's the joke? What's the deal with the breathalyzer for that? So of course it prompted me to take a look, and here's the breath test developed by Indiana University researchers. This one, collaboration with Texas A&M and the Air Force, looks like a kiosk, and you blow into this big thing, and it sends the results to your telephone. This Israeli company developed a breathalyzer with 98% accuracy. You can see the healthcare worker here administering the test, and I'm not sure how well it shows up on the slide. She's reaching through this plexiglass panel with her two arms in order to cut down the likelihood at all. And finally, I just looked it up on Wikipedia, Alyssa, and it basically said the breathalyzer is a diagnostic device enabling the user to determine with 90% or greater accuracy the presence of coronavirus in an exhaled breath. It's what the dogs detect at the stadiums. As of the first half of 2020, the idea of a practical coronavirus breathalyzer was commonly developed by unrelated research groups in the United States, Netherlands, Finland, Germany, Israel, England, Australia, Indonesia, and Canada. So if you'd like to take a look at any of the others, just check out Wikipedia. So are there any questions that are related to the serious part of this presentation? That last slide, Melissa, is not, Alyssa is not 508 compliant, okay?
Okay, that's right. That was interesting. That's funny. I did think that wooden square box was a joke. So now I learned something. While we give participants a chance to enter questions into the chat, I actually had a question. I think I heard you say, and correct me if I'm wrong, that HIV-2 cases are reducing, are becoming increasingly rare. Is that?
No, they've always been rare, actually.
They've always been rare.
No, we didn't know because there were some in New York, and some of the tests, like the Western blot, didn't pick up HIV-2. And so, back in 2010, 11 years ago, when I had actually first proposed that algorithm with HIV-1, HIV-2 differentiation assay, the real goal was to get rid of the Western blot at that point in time. And the HIV-1, HIV-2 antibody test that worked real well was that the multi-spot rapid test that a bunch of labs could do in 15 minutes. So it was real easy. It made sense to do it as the antibody test. There was I don't remember the name of the paper from CDC, but it basically sort of said, not worth the squeeze, screening for HIV-2. There's a lot of effort and very low yield for doing that. So I don't think that there's really much point anymore in screening for it. There is now, however, and I should have mentioned that as well, an HIV-1, HIV-2 RNA differentiation assay. So for the people who are down in that bottom little low corner where this is negative and that's not, they might have HIV-2 and they're indeterminate, you can actually get HIV-1, HIV-2 qualitative tests that differentiates between those from Roche.
Interesting. Well, that was great. Well, looks like we don't have any further questions. So that concludes our presentation today. Thank you so much, Dr. Branson, and thank you to the Arizona Telemedicine Program. And again, just notice that after you exit the webinar, you'll get a evaluation screen for the telemedicine program. If you could take a moment to complete that, and then we will also be following up with our own evaluation email. We appreciate it.
Great. Thanks to everybody. I hope you enjoyed it.